This has been an astonishing 3 days for me to be able to go to several different places, talk to many students, many faculty members, and get the opportunity to just talk, which I love to do. And it's been a real privilege. I've often said to people that one of the prizes that comes with the Nobel Prize is that for some reason people invite me to give general talks about science and don't want to know about the intricacies of our latest genetic experiments. But we all too often don't give that opportunity to our scientists to talk more generally about what they do and how they feel about things. So it really is a gift that comes with the Nobel to be able to come up and talk more generally. And that's what I'm going to attempt to do today. I did notice, though, in the last few years since the Nobel, that I realized if people were going to invite me, they wanted to at least listen for a couple of minutes, and I could say whatever I wanted. And so I've decided to add advertisements to my lecture. So I have a variety of these advertisements, so I'm only going to talk about one. And they're usually an advertisement that has a request to it, and you'll see what the request is. One of the wonderful consequences of the Nobel is that I had been asked about 4 years ago to be a member and then the chair of the Committee on Human Rights of the U.S. National Academies of Sciences, Medicine, and Engineering. I thought I would only be working a couple of days a year. I actually do something about it every day, which is a sorry statement about the problems of human rights all over the world, but it has been greatly rewarding. Most of the time we work on behalf of scientists, engineers, and health professionals anywhere in the world whose human rights have been greatly abused. But in recent years we have done, and that's all behind the scenes, but in recent years we have become more and more interested in outreach and bringing more people in to help us in these endeavors. And so last year earlier this year, we changed our website and made a poster. And that's what's shown in this next slide, of 8 things people can do to promote human rights in science, engineering, and medicine. And this poster, as you can see, there are a variety of things here that one can do. This is also the portal in our website to many pages on the background material and information on these various aspects. And if you look in the upper right-hand corner, you see host a discussion on human rights and/or integrate human rights into your teaching. I would like, this is the request part of this, I would really like people to get in touch with me if they have ideas about how human rights can be used in the teaching of STEM subjects. We're trying to amass a very large library of these things. I'm going to give you one example of what I have done in the past to give you an idea about what I'm talking about. But that's the request. That's what the advertisement is about. If you have ideas, please let me know about them. How can you let me know? It's a little hard to see the URL at the bottom, but if you just go and look for nationalacademies.org/human rights, you will get to the page of our public website. You will see a picture of this poster. You can click on that. That'll bring you into the portal where all this information is, as well as a way of downloading the poster. You can print it up and do what I do, which is to put it up in my lab. So what is an example of how one can use human rights in the teaching of science? Well, I'm a geneticist, and as I'm sure almost all of you know, you cannot teach genetics without talking about Mendel. He's the first person we talk about, and we just bore people silly talking about peas. And we talk about the general principles, and people start the class, and by sort of near the end of the class, people start to doze off knowing that this is peas. It's not really that important. And that's when I bring up something that was very important. As some of you may know, in the 1970s, the government of Argentina was run by the military, and it was a very sad time because many people in their early 20s, people like the students here, were disappeared, taken off the street, executed, and put into unmarked graves. People were picked up as families with young children. The parents were killed, the young children were given off to other people. In some cases, pregnant women were picked up. They were allowed to give birth, their babies given away, and the mother was killed. After the Falkland War, the military junta was deposed in Argentina, and a group of the parents of the disappeared got together, and they wanted to be reunited with their grandchildren. They were called the Abuelas of the Plaza de Mayo, the grandparents, the grandmothers. And they wanted to know how they could be reconnected with their grandchildren. And a series of scientists, including Mary Claire King, who's now at the University of Washington, went and tried to figure out how they could deduce whether a child was actually part of a family and it had been the child of disappeared parents or not. And they were able to work it out. And there's a wonderful article by Jared Diamond that came out in Nature in 1987. It was a News and Views. You can see the reference on the page, in which it shows a family. If you look at the sort of next to last line, you see the first two individuals, Claudio Ernesto Lagares and Monica Grinspoon, have a line through them. They were disappeared. But there were their siblings, or Monica's siblings, there were Claudio's parents, there was the grandmother of Monica, and together they could deduce, for one genetic locus that had an enormous amount of variability, they could deduce all of the contributions of the parents, and sort out from that what were the possibilities for this child. Well, in the court case, they presented this and said that this child indeed had the situation, had the genes that could have come from the grandparents. The so-called parents, the people that she had been given to, refused to be tested. But they were able to show in court that this possibility would come up from those grandparents. That was a likely thing to happen. But if it happened just by chance, it would be less than 1 in 1,000. That's more than enough for a court case, for the court to do it. And the judge gave custody of the child, who was then 8 at the time, Paula Eva Lagares, to her grandparents. They took her home. She immediately went to the room she had been in when she was 2 years old and picked up a doll that had been her favorite doll at that time. This has been done now with many people, but all this uses is simple Mendelian genetics. And so when the students hear that, they're a little shaken up, which I believe they should be. But they also realize that what we're doing in science has implications in other things, and we can use it in inventive ways. So I invite you, if you have ideas, please send them to me. I hope you get interested in our website as well. And now I'll give the real talk. So when I was growing up, I heard a lot of stories about famous scientists: Galileo, Newton, Darwin, Einstein, all these wonderfully inventive people. And hearing these stories, I had an idea about what it meant to be a scientist and how scientists act. I'm going to tell you what those myths were, and then I'm going to tell you about the GFP story, which says that all of that was nonsense. But I think I should start with the myths. So the first myth is that scientists are geniuses. And if you're not a genius, and the first B you get on an exam proves you're not a genius, I have many poorer grades than that. I won't go into my transcript. Then you shouldn't be a scientist, because scientists are geniuses, because those are the stories we tell people about. Amazingly accomplished people. The second myth is that scientists' experiments work all the time, because we don't have time to tell you about the failures. We just tell you what happened. And if you're working in a lab and it doesn't work instantly, that's another proof that you shouldn't be doing science. The next myth about scientists that came from all these sciences is that scientists think differently from everyone else on the planet. They come up with this peculiar thing we teach grade school kids about called the scientific method, where they're sitting around, they come up with an idea, they come up with a hypothesis, they think about that hypothesis for a while and devise the perfect experiment. It works the first time because they're geniuses, and they get a lot of money and prizes from that. But it's purposeful, nothing accidental about it. And another thing about all these scientists, now I have to tell you, I took high school biology in 1962. That was the year that Watson, Crick, and Wilkins won the Nobel Prize for the structure of DNA. Before that time, and that hadn't made it to our textbooks yet. It wasn't there. So everyone I had heard about, all these great scientists of the past, they were loners. They just were entirely on their own, although for some reason they could have an assistant named Igor. But they were on their own. People did not work in groups. And finally, all these great scientists in the past, except for some very rare exceptions, Marie Curie, George Washington Carver, they were all white men, Europeans or of European descent. This last point, I think I want to make another couple of comments about. If you go online today and ask for most cited scientists and look at who's portrayed as a famous scientist, you get this group of pictures, which seems to say we haven't changed much since 1962. In fact, in this picture, only Marie Curie at the top and Rosalind Franklin at the bottom are the only non-European or European-descended white males. You have to go on to another set of pictures before you get anyone that's not of European descent. In contrast to that, I want to show you a picture of the people that have come through my lab and worked in various jobs in the lab over the last 7 or so years. And as you can see here, and although it says technician on the top, most of those people went on to graduate school or on their way to going to graduate school. They come from Chile, Canada, Peru, Colombia, China, Iceland, the Gambia, Greece, Vietnam, Israel, all over. Actually, now we have someone from Haiti, or his parents came from Haiti. We have people from all over the world. They all contribute. They all bring something unique to the experiments. I contend that this is what a modern lab looks like today. It is nothing like those pictures on the previous slide. So let me show you why the GFP story says that this is all a lie. Now, my involvement in the GFP story started in 1989 when I went to a seminar. But I have to tell you what I was interested in when I went to sit down in this seminar. It was a lunchtime seminar. I could get a free meal. It seemed like a good thing to go to. And it was about neurobiology, which is what I was interested in. But what I was really studying at that time was a set of nerve cells that were the cells that sensed gentle touch in the nematode Cenorhabditis elegans. Here's a picture of the nematode. And the one thing, I'd been working on this animal and doing genetics with this animal for something like 12 years. And at every seminar, I would bore all, especially the Drosophila geneticists, I would bore them by saying all the wonderful things about the animal. It grows in 3 days from egg to egg-laying adult. We know every cell in the animal. We can do genetics up the wazoo. It's only 3 days for generation. It's great, and it is transparent. Well, at this time, we were in the middle of cloning a lot of genes that were needed for touch sensation or for the development of these touch neurons. And one of the questions we wanted to address was, what cell activates that gene? And this would be a great story if I was able to tell you that no one ever thought of that problem before. But that would be a complete lie, because people have been thinking about that problem forever, and they knew how to answer it. If you want to know where a gene is turned on, look for the products that come from that gene. So you could make an antibody and see where the protein was made. Or you could use, on the bottom, a probe to look for the RNA that's made by in situ hybridization. Or you could use a reporter, and the most common one, Malcolm Casadaban came up with in 1962, which was using the E. coli beta-galactosidase gene, LAC-Z, and having expression of that enzyme, and using an appropriate substrate called XGAL. Whenever that enzyme was present, you would get a blue precipitate. And so you could see exactly where the cells were that were expressing the gene by using this reporter. So it wasn't a problem that we didn't know how to do this. We had already done all these experiments. But there was some problematic aspects of this. One of the problematic aspects was that in order to do any of these experiments, we had to prepare the specimen, which meant killing it, fixing it so things didn't get jumbled up, and then in order to get the antibody into the animal, or the substrate for the beta-galactosidase, or the probes for the in situ hybridization, we had to permeabilize it. So we were looking at dead tissue. We were just getting a static view of what was going on. We had no real idea of what was happening over time. And so that was a problem sort of in the back of our heads, but not prominent. It's just we couldn't do that. I want to explain the color code for people that you're going to see. Anyone whose name, as these two people are, in red are people from my laboratory. Anybody whose name is in blue is a collaborator. Anyone whose name is in black did something I wish I had done. And I may as well start with that, because this seminar I listened to, the beginning of it, the speaker talked about the work of Osamu Shimomura, who was one of the three of us that won the prize in 2002, and the remarkable experiments he did. And it was astonishing. Actually, he's astonishing. I urge you all to go to the nobelprize.org website and read his speech. Read about what he had to go through. At the age of 16, he's told he has to quit high school and work in a factory. He leaves the school, he works in a factory which is 10 miles out of the town he's in. Turns out that's a good thing, because while he's in the factory, he's temporarily blinded but protected from the bulk of the force of the atomic bomb that destroyed Nagasaki. He goes home, he attributes his survival of the whole thing to his grandmother, who takes one look at his white shirt and sees that it's completely gray with radioactive ash that had fallen on his way home, tells him to take a bath, and he attributes his survival to that. He goes and helps people. Can't go to college. There is no college. Eventually, they build up the pharmacy school. He can go there. He said he wasn't interested in the pharmacy school, but it was the only college he could go to. He goes and works as a technician. And through, I won't go into it, a sort of an accidental discovery, he enables the lab to work out how a crustacean, a small crustacean called Cypridina, actually generates light. It's a bioluminescent organism. There are many bioluminescent organisms, but they all work differently. Glowworms, fireflies, bioluminescent bacteria, jellyfish, fish, but they all do it differently. It's been estimated that there's something like 40 different biochemical reactions that generate light. Most of those have not been elucidated. And so he figured one out as a technician. From that, he gets an invitation and a fellowship to come to the United States to work on other bioluminescent organisms. And his boss at McGill University realizes he will be paid double if he has a doctorate. So that's his going away present. Not a bad one. I'm sort of envious, I must say. And he comes to the United States and he starts working on this jellyfish, which produces a beautiful green light. And this is where the experiment's working every time in the scientific method. This is going to be questioned here. Because every time he grinds up the jellyfish, he can't get any light out of them at all. The experiment is a complete failure, time after time after time. He bribes his kids to find more jellyfish so he can experiment on them. Nothing works. Finally, one night, towards the end of the summer, he is frustrated once again. And he decides he wants to go home for dinner. He's going to clean up, takes all the samples, he throws them in the sink where there'd been some seawater and some other things. And he turns off the light, and he's about to leave the lab when he looks back at the sink and is startled by the fact that it's glowing brightly. So he stops and he looks at this and thinks about it, because nothing was glowing brightly before, and realizes there must have been something in the seawater that triggered the production of light. And reasons, well, I'd never had calcium ions. So goes back eventually and does the experiment after the next couple of days, and sure enough, adding calcium triggers the production of light from the solutions he has. He uses that to purify the protein, which he names after the jellyfish, a quarin. This actually turned out to be the first calcium indicator that anyone had. People used to inject a quarin into cells and see what happened with changes in calcium. But when Shimomura looked at this, so let me just point out, I know of people that have had very successful experiments by throwing their preps on the floor, picking them up and redoing them, or on the lab bench. It's not exactly the scientific method, but it sometimes works. In any case, he purifies this protein. He has the bioluminescent protein. The only problem, it gives the wrong color. It produces blue light, not green. And I imagine there was a fair amount of swearing at that point. And he says, well, maybe there's something else. Maybe there's something that converts the blue light to green light. And so he takes a handheld ultraviolet lamp and he tries irradiating all of his samples. And sure enough, there's one that if it's irradiated by ultraviolet or blue light, it gives this bright green light back. In 1962, he writes up his experiments. Most of the paper is about a quarin and the studies he did with that. Footnote number 3 says, by the way, there's this other protein. If you shine blue or ultraviolet light on it, you get green light out. I'm going to call it the green protein. That's what he won his Nobel Prize for. So you don't have to write a lot to get a Nobel Prize. We, of course, don't call it the green protein anymore. We call it GFP, or green fluorescent protein. And that's what I heard about when I was listening to the seminar. I had known about a quarin. I had never heard of GFP. And especially when the person giving the talk, a man named Paul Brem, said, you know, GFP, it's just the protein sequence. You don't have to add any cofactors. There's nothing else needed. The only thing you do is add light, and you get green light back. Add blue or ultraviolet, you get green. And I got really excited because it's a molecule that you can visualize simply by shining one color light in. And if you work on a transparent animal, you are set for life. So I got really excited. I thought of all the experiments I would like to do, because now, if I could put this in the worms, I could look at it in living animals. I was very excited, so excited, I have no idea what the seminar was about, because all I did was fantasize about the experiments that we could do. The next day, I got in touch, because I realized that this was really providing a lantern to visualize things within cells. I got very excited. I did what any head of a lab would do under those circumstances. I got on the telephone, and I got in touch with this man, Douglas Prasher, who I found was, in fact, trying to isolate the DNA that encoded GFP. And I was very excited about that, and he was excited, and we were going to do this great experiment together, and then we lost track of each other for 3 years. Finally, a new graduate student, Gia Skirkin, came into the lab, and I told her that I had this wonderful idea. All of my ideas, I feel, are wonderful. And I talked her into trying to, I said, I'd really like to try this experiment, but Douglas has not gotten back to me. And then we found that we had just lost track of each other, and he actually had succeeded in finding the cDNA for this. Didn't think it worked, but we called him up on the telephone, and he was very happy to send us the sample so we could try it. And so Gia's project, as a beginning graduate student, was to see if she could put the gene for GFP into bacteria and see if they would glow green. And as you can see from the slide here, in the picture on the lower right-hand corner, she succeeded. This was the experiment that worked the first time. She put it in, she was able to get it to work, but not without some difficulties. And the difficulty, she had worked previously to coming to my lab. She had gotten a master's in chemical engineering from our engineering department at Columbia. And she had worked on fluorescence, which was one of the reasons I really tried to twist her arm to do this experiment. And she did this experiment, put the sample under our microscope, and immediately realized that our microscope was a piece of junk. So she went to her old lab, and that's what it says here on this part of her lab notebook page. And she said, she looked at it, immediately it was obvious the bacteria were glowing green, and that's when she took that first picture. We did a lot of jumping around then. We were very excited about this, but this gave us a big problem. The problem was we didn't have a usable microscope. And the people over at engineering were not really happy with the idea of us coming over all the time and using their equipment. So I had to find a microscope, and I called up all the microscope sales representatives in New York City, and I said, we've just developed a new way of looking at gene expression. It uses fluorescence, and we're going to have to buy a new microscope. We just don't know which one that's going to be. But we'd be able to make up our minds if they could bring a loaner. And if they could bring that loaner and let us use it, we'll give it a good workout in maybe a month or two, we would be able to do this. And so, in fact, if you look in our paper, you'll see that there's a footnote that says, the following microscope, you can see GFP very nicely in all these, those are all the loaner microscopes that either different people or different companies let us use while we did the experiments, and then afterwards we did it. Now, I said Gia's experiment worked the first time. This was a big surprise to everyone because no one believed the experiment was going to work. In fact, if you had asked anybody in the field, should we do this experiment, they would say, no, you're being idiotic. And the reason they thought that was a very funny thing that happened in the molecule. So I'm sure you all know proteins are linear chains of amino acids that are connected by what's called the peptide backbone. It's a string of atoms. And GFP starts like that. You can see here where it says SEER65, it goes up, that oxygen, nitrogen, goes around, another oxygen, and out to the right, it's a chain of atoms. But in GFP, that portion in the middle, those 5 atoms, come together to form a 5-membered ring. Not anything that anyone had seen in any other protein. And people didn't have a clue as to how that was made. But they were sure it was going to require 1, 2, 3, 5, some number of converting enzymes that would allow for this chemical reaction to take place. So the idea that you would just put the gene for just this protein in was ludicrous. But we tried it anyway. It worked. It worked the first time. This was great. It said there are no converting enzymes, or more particularly, it is its own converting enzyme. It allows this reaction to take place. And so it worked on its own. If it had needed all those other things, it wouldn't have been as useful a tool, because you only want to have one thing that you're looking at. So it was great that it worked the first time. But I found that there were 3 other groups that were trying to do the same experiment at the same time. They failed. We did not. And the reason they failed, I believe, and we did not, is because they were careful and we were sloppy. Or at least careful in a different way. The DNA that Doug Prasher gave to all 4 groups can be diagrammed here. The green part is the part that encodes the amino acids for green fluorescent protein, whereas the red parts are just extra jellyfish DNA that happen to come along for the ride. Now, at the time, if you wanted to be careful, you hired an expert to make your DNA for you. That expert was called E. coli, because the bacteria could really copy accurately DNA. And if you did that, you would have to then take the DNA and use the portion you wanted, and the only way to get the jellyfish gene out of this was to cut it with an enzyme called EcoR1, which of course gives you a piece of DNA that is both the red and the green parts. For reasons people still do not understand, that does not work. We didn't want to do that. I was just very scared of anything that wasn't what we really wanted. And so we used polymerase chain reaction, a way of amplifying the DNA, that we could just amplify the green bit. Now, the problem with PCR is that it made a lot of mistakes when it copied the DNA. So a lot of it was just no good. But I reasoned, we want to put this into E. coli. We don't want to put it into 1 or 100. We're putting it into millions of bacteria. And if we're putting it into millions, I don't care if 99.9% of it is garbage. I just want a small amount so I can see some fluorescent bacteria. And that's what we got. So we were sloppy, maybe careful in a different way, because we excluded the thing that other people inadvertently found killed the reaction. So with this, and then we put it into worms, we decided we wanted to publish this paper, share the information with people. And at the time, the journal Science was the journal that had the largest subscription base. And so we sent it to Science. It did get published. I was very happy eventually about that, but not without some problems. So very quickly, problems in publishing. First, we sent it in. They don't just send it to reviewers. They ask, is this a paper worthy enough of science that we should consider reviewing it before rejecting it? And they called me up and they said, no, we're sending the paper back to you. And I said, what's wrong? What is wrong with the paper? They said they actually liked it, but they weren't going to do it. And I said, why not? And they said, well, we don't like your title. I said, what's wrong with the title? Green fluorescent protein, a new marker for gene expression. And they said, look, everything in our journal is new. You cannot use the word new in a title. I should tell you that I do not like to be told what to do. Maybe that's why I'm a scientist. And I think you can tell how aggravated I was by the fact that I resubmitted the paper with a different title. That title was, the Acquaria Victoria green fluorescent protein needs no exogenously added component to produce a fluorescent product in prokaryotic and eukaryotic cells, which is a complete description of the paper. Excuse me. There is some water here. To my great joy, the paper got accepted. The copy editor then called me up and said, do you know that your title is very long? And I said, oh, well, maybe I could change that. Oh, please. Yeah. And so the official title is now Green Fluorescent Protein as a Marker for Gene Expression. And that's what you'll see in the journal. The second problem was the fact that we had this picture, and that cell that starts with a blob and ends with this sort of extended fingery thing. That's called the growth cone of a nerve cell. This is a picture of a living animal, and I wanted it to be on the cover if at all possible because it was going to show people that they could look at living organisms and follow development or any other thing they were interested in. And I sent it in, and I get this call from the cover editor, and cover editor says, we really like your picture, we want to use it on the cover, but we hate putting anything on the cover that's green. Can we change the color? And I said, no, you're not going to do that. And it actually does. This is a very enhanced picture of it. It doesn't look so great on the cover, but I just couldn't have them change it. And then the final thing was, I basically can't keep my mouth shut. And that means I had already told lots and lots of people about this. And people had already gotten samples from us to use in their own experiments. And people had even gotten back to me to say, you know, it worked for us too. And I said, can I use that information? We'll put it in a footnote, your unpublished data. And they said, well, you gave it to us before you published. Of course you can. Except for one person who was very problematic. This person. You can't read her letter, but let me tell you that in order for me to be allowed to publish her or talk about her work, I was supposed to make coffee every Saturday morning for 2 months, prepare a special French dinner, and take out the garbage nightly for a month. This is my wife. But what Tula did was a truly spectacular experiment. You know, genes basically have two parts. They have the coding part that makes the product, and they have the part that says where, when, and how much, the regulatory part. And what we had done is taken the regulatory part and have it drive the production of the GFP. What she did was make the first protein fusion. She had the regulatory part, the protein coding part for the protein she was interested in, and then GFP. And the protein she was interested in was a protein that was made in fruit flies. We have a lot of family arguments of worms versus fruit flies. But she was interested in this protein, which you see here in green because it's got GFP attached to it. That protein is made in the cells called the nurse cells on the left-hand part of the diagram, but is then transported in to the developing oocyte to lie in very specific regions. And she could watch the migration of these proteins from one cell into the other. It was a spectacular result. That letter never got sent to Science. That was her letter to me. It was a very nice letter. She said, yeah, okay. GFP has many advantages, and people have taken it up. All you have to do is put the DNA encoding it in, and it will work. It doesn't hurt the organism to have blue light shown on it, so it's not that hurtful to it. It's a small size, so it goes everywhere in the cells. And as I've already said, you can see living organisms. And here's some mugshots of different organisms, Drosophila, actually at the top nematodes and Drosophila, then Alba, the GFP bunny, canola plants, mice, zebrafish, and a bunch of cells. And you can see that for that Purkinje cell on the lower right-hand corner that it really has gotten all through the cells. But let me show you a couple movies made actually here in Canada by someone I've never met, Rosalind Silverman Gavrilla. But fortunately, she put these films on the website for the American Society of Cell Biology, and I think they're really quite wonderful. Both of them are movies made of early Drosophila embryos, and they have different things that are attached to GFP. So if you look in the center, in cell division, the chromosomes come to lie on the spindle. The spindle is made up of microtubules, and the movie on the left is a movie of several rounds of nuclear division in the embryo, 4 in fact, in which the microtubules have a protein, tubulin, that has GFP attached to it. So you're going to see this, let me just start it. You see the spindles forming, and it's very much sped up so we can see 4 rounds. And I think the thing that's really remarkable about this, and in fact is seen in the name she gave this movie. She calls this movie In Synchrony. Because as you look at this, one of the remarkable things is all these nuclei are dividing at the same time. And immediately, by looking at the living prep, starts to invite questions about how is this coordinated division taking place. On the right-hand side, we have a falsely colored image that is an image of nuclei. GFP is attached to something called a nuclear localization, so if there are nuclei, it's in them, but when they break down, it's all over the embryo, and then the nuclei reform, it's going to be sucked back into them. And there you see it going away. Now it's everywhere in the embryo. The nuclei are going to reform, and the stuff's going to be sucked into them. She calls this Starry Night. I think you can understand why. But here you see that it seems to go in a wave from one part of the embryo, one part of the field to the other. We think that's because there's some slight damage to the prep. But this says we have a very nice way of detecting slight damage to the prep. There's a book in the United States and maybe here for kids called If You Give a Mouse a Cookie. And you give the mouse a cookie, it wants a glass of milk, you give them the milk, wants a straw, after the straw, wants a napkin, it just keeps going on and on. That's what scientists are. And the person that gave us many of those things, the milk, the cookie, and everything in GFP, I'm going to fast through the picture of that, is this guy, Roger Chen, who got the first differently colored GFP, made a number of other spectacular improvements on this, and was the 3rd person to share the Nobel Prize with us. And among other things, as was mentioned before, these many colors were used by Josh Saines and Jeff Lichtman to make this wonderful way of looking at nerve cells. It's been used in Drosophila, in zebrafish, and it started off in mouse, looking at the central nervous system. And if you have something with different colors looking at the central nervous system, there's only one name for that, and that's brainbow. And these are some of the really spectacular pictures. Now, GFP, like any development in science, started off as a basic science observation. It leads to more understanding of basic science, more applications, and some really unexpected things. So very quickly, what are these things? Well, one of my favorites is some work done by Tony Hyman and Cliff Brangwyn in 2009, in which they used GFP to mark a protein that they thought was in the cytoplasm, only to find that it seemed to be in this fluid that acted as if it was oil in water, that it was separated from the aqueous part of the cytoplasm, even though it was known to be an aqueous protein. So it was a very strange and quite wonderful observation, which is called a phase-separated particle. No one before that time had any idea that that was what was happening in cells. We now know that many RNA-containing particles act exactly like this, both in the cytoplasm and in the nucleus. It's a completely new way of looking at the cell. Absolutely spectacular discovery. It's been used, GFP has been used to look at how the AIDS virus is transferred from one mouse cell to another, and here's a movie of that. You see the AIDS particle, the virus particle attached to the cell that was infected because it has GFP, and you now just saw that it was distributed into another cell. Now, why is this important? Well, most of the time when we teach introductory biology to people, we talk about cell lysis, phage, infecting cells, cells exploding and going everywhere. Well, if a cell explodes, then antibodies could bind to things. But if it just goes from inside one cell to inside another, you got to think of another way to stop that transfer. So being able to watch what was happening in these cells in culture, I think, can be very important in thinking about human health. And then there are some very unusual things. One of my favorite is Bob Berlage made a bacteria in which the explosive TNT would trigger the production of GFP in the bacteria, which you might think is very strange, but he knew that landmines leak leak TNT, and and what he was looking for was a way of detecting leaked TNT from landmines. He had a friend bury 5 active mines, disconnected, in a field, and sprayed the bacteria on it, came back at night and took a look, and was able to find where the landmines had been buried. Several other groups have repeated this experiment. The problem is, it's not ready. And it's not ready, even though it would save, I would expect, many lives of non-combatants, because you never want to have a false negative. And people are still working on making it 100% correct. But there's several groups doing it. GFP is used in other unusual ways, been used to make silk that's fluorescent. And then it's also been used by screenwriters particular screenwriters and directors, Ang Lee, to give the green hue to this person at the beginning of the movie. It was quite nice. So let me finish up with a couple of things. The first is, I want to talk a little bit about those things I started off with, about scientists, all that stuff about the the things I had learned. So the first thing is, scientists are geniuses. I showed my transcript at a college reunion after the Nobel, and everyone came up to me and said afterwards, "Thank you for showing that. I was embarrassed about my transcript." But Shimomura was really a spectacular scientist, developed into one under amazing conditions. Roger Chen won the highest prize for high school science students in the United States when he was a high school student. So he was showing amazing promise very early on and continued to do many, many wonderful things. So I think the answer to this thing is that some scientists are geniuses, certainly not all, and none of them all the time. Okay? I won't go into what I mean by that. Scientists' experiments work all the time. Well, I told you Shimomura's experiments, which never worked until by accident he found to add calcium. And our experiments did work all the time. So that, I think we would say, is rare. Scientists use the scientific method. Yeah, not always. But there's there's a wonderful quote from the physicist Enrico Fermi that goes something on the order of, if you do an experiment, it confirms your hypothesis, you have made a measurement. If it doesn't confirm your hypothesis, you've made a discovery. So as I say here, I think the scientific method, using the hypothesis, comes after the initial discovery, the realization that there's something unusual that has happened and that you have to follow it up. So it is a way of thinking about things, but it's not what starts it. The discoveries are what started. How do you get these discoveries? You have to do a lot of experiments, and hopefully we all keep our eyes open to see what is unusual and what's something we'd like to pursue. The next point, scientists work alone. Well, here are all the pictures of the people that were involved in our paper on GFP, showing that it was a marker. There were 5 of us. It was a team effort. It's always been a team effort. It's not an individual thing. In fact, GFP has sometimes been used with other proteins, other fluorescent proteins. So you'll have a GFP and then a YFP, or rather, usually a blue fluorescent protein and a yellow fluorescent protein. The blue is excited by ultraviolet light, and if it's close enough to the other, it doesn't even make blue light. It just gives its energy to the yellow fluorescent protein, and now you get yellow. Or you can imagine a whole chain of violet to blue to yellow and so on, to red. I think that's a good metaphor for science. that we in fact take the ideas and observations of other people, add to them our own experiments and observations, and change what they found into something different that will then be taken by somebody else in turn to change things again. That really science is a cumulative process, not one of individuals making great discoveries. Everything in my lab has been work by a team of people, and it's been more fun that way. So, do scientists work alone? I'd say rarely. And the final one, science is done by white men. Well, that's just nonsense. Now, I have a whole list of other conclusions, but I've already been shown that I have no time left, so I'm going to skip these and just really get to the ones towards the bottom. I think the story of GFP says that all life should be studied. Unfortunately, a lot of effort is put, and it's good effort, I'm not against it, but there's a big emphasis on what are called model organisms. I don't like that term because I think it emphasizes their model, presumably for human disease. But in fact, they're the ones that teach us about things we should have been looking for that we had no idea about in human biology. RNAi, cell death genes, all sorts of this phase-separated particles that may actually have an implication for Alzheimer's. All of these things were never even imagined in human health, but were done by studying other organisms. So I don't call these things model organisms because they teach us so many new things. I refer to them as pioneer organisms. And the final point I want to make is that this work says that fundamental or basic research is really essential. It's what starts the pipeline that leads to applications. But you can't just stop it and expect there to be the applications that you want. We are still woefully ignorant about biology, about human biology. We don't know the basis of the diseases because we don't know the fundamental biology that could be affected. There is so much more for us to learn, and it's getting that pipeline, continuing that pipeline of new information leading to applications important. So both are important. And I want to leave you with my favorite quote about basic research. It was said in 1969 to a joint committee on Congress trying to debate whether there should be money set aside by Congress during what was then called the war in Vietnam for this enormous science project, this particle accelerator that was built called the Fermi Lab. And they asked Robert R. Wilson to come to Congress and explain why in the world taxpayer money should be used for this particle accelerator. And one of the senators, John Pastore, who wanted this to happen, thought he would give this physicist a very easy question. He said, Dr. Wilson, please tell the members of the committee all the ways, how many ways are there that this particle accelerator will help national security? Wilson said, none. The senator did not want to hear that. He asked it again and again, and finally he said, look, Dr. Wilson, please tell the committee, in what respect will this particle accelerator help the national defense? At which point, Wilson said the following, although I think he may have prepared it ahead of time. It's too good. He said, it is only to do with the respect with which we regard one another, the dignity of men, our love of culture. It has to do with whether we are good painters, good sculptors, great poets. I mean all the things we really venerate in our country and are patriotic about. It has nothing to do directly with defending the country, except to make it worth defending. Thank you very much. It's always surprising when the slides are sort of bad, but they were good yesterday, so I apologize.
Serendipity. Is there somebody waving their hand? There must be somebody who wants to ask a question. Don't be shy.
You won't have to take your exam if you ask a question.
Thanks very much for the talk. It was really great, and I agree it's really important that basic science is funded. I have two questions. The first is, I'd just be interested, because when else am I going to get an opportunity to ask this, your perspective on how the Nobel Prize is awarded. Obviously, it can only be awarded to 3 people, and it seems like there are a lot of other people involved in the discovery. And the second one is about your assertion, which is definitely true, that science isn't just done by white men. But how do you think at this point, is it just a game of numbers, that in the future the people who are honored for scientific discoveries are less likely to be white men just because there are more people who aren't white men doing science? So just
I would
interested in
certainly
your
hope so
perspective.
for the last. Tell me the first question again.
Oh, just what your perspective on how the prize
so the
is
Nobel
split.
is restricted to 3 people. They never ask you. You never know you're being considered. So I can safely say I have no idea. As you listen to this talk, you heard of at least 6 people. Shimomura, Douglas Prasher, myself, Gia Skirkin, my wife Tula Hazelrig, Roger Chen, all of who contributed exceptionally important aspects to this story. Now, as I said, I'm not asked who they do. I imagine, I always think about the Nobel Prize as a pie that can be cut in many different ways. And it must be agony for them to come up with the decisions that they do of who should get the prize. I could easily think of this prize in 2008 having gone, for example, just to Osamu Shimomura for discovering this incredible protein. And everything else that everyone did all came out of his discovery. Roger Chen, before the work on GFP, had done spectacular work using fluorescence to measure things in biology. And one could have imagined that since this was a chemistry prize, and he was very much a chemist, that they could have given him a prize for using fluorescence in biology. They could have decided that the prize could have gone for actually showing that you can use GFP as a tool, and that would have been me and Douglas Prasher and my wife, or Gia Skirkin and me and Douglas. It's a hard one to decide who the 3 are going to be. I would have loved it if my wife had been part of it. But that's a decision that must be agony for them. It is very unfortunate that, Sarah, I'm, however, very grateful that they decided I would be part of this story.
The second question was about the change from white men.
So the answer to that is we have still an enormous amount to go. I tell with some trepidation the fact that when I joined my department at Columbia in 1982, the percentage of tenured track or tenured faculty that were women was about 25%. And although the department has grown quite a lot since then, the number is now around 28, 29%. That's not any good. That really needs to be changed. We're seeing a great deal of very talented young women in college. It's about 50-50 for majors in biology, the same for graduate school, the same for postdoc. It's afterwards that the number goes down, and we really do have to work on that. And I think there are ways that one can do it. We certainly are not at a point of sitting back on our laurels and saying, okay, well, the pipeline's working, we're going to be able to get all this through. There's still a lot of work to be done.
Thank you. We have at least 6 questions queued, so let's go.
First of all, I use GFP and its derivatives on a regular basis, like I'm sure a lot of people in this room do, so thank you very much for helping provide it to us. But my question's about how a lot of science tends to happen concurrently, and you kind of, how a lot of scientific discoveries tend to happen by multiple labs or are kind of in progress by multiple labs at the same time, and you kind of alluded to that a little bit. And I'm just wondering if you could tell us a little bit more about the history in terms of what other initiatives were ongoing at the time while you were writing the paper. I assume it took you a little while to get the gene into the worms and other organisms, and also going forward, as Roger was doing the mutagenesis, were there other groups working on that? Were you guys working on that? I'd just like to get a little bit of the story behind it.
So the answer is, I didn't know that these people were doing the experiments. I tend not to know that other people are doing things, and so it wasn't an issue with me. I didn't think about it. That's just the truth of the situation. I did my postdoctoral work in the lab of Sidney Brenner. And Sidney always liked to say that he was really good at coming up with new things. Not so good at following them up because other people could do that. He was really good at coming up with new things, and he really liked coming up with new things. He also said, don't ride the wave of popularity. You should always be a half wavelength off. You just will never know if you're a half wavelength ahead of everybody or a half wavelength behind. But in any case, there was this idea of starting things that other people were perfectly capable of continuing on. And we had been doing other experiments on nerve cell development and function beforehand. We went back to doing those, which I thought would be sort of innovative.
So where's the microphone got to?
Thank you very much for a wonderful talk. You speak a lot about what scientists are not, and I was wondering if you could let us know what you believe a trait which truly successful scientists have.
So it's somewhat difficult because I think people are very different and they bring different perspectives and different attitudes. I've seen people that are amazingly efficient in what they're doing, very directed in their science. They do great work. And there are other people that use the lab as a clubhouse. They're there all the time. Their friends are there. Every once in a while they get to an experiment. They also do great work. There are, one time soon after my wife and I were married, both being scientists, we were talking and I get my ideas usually by talking things out with people or diagramming things. I don't get them by sitting quietly and thinking. And my wife one Saturday announced she was going to go in a room and just quietly think about things because she needed to come up with a new experimental system or new series of experiments. And I just thought, that's the most ridiculous thing I ever had. And of course, she went in the room. 2 hours later, she came out. She said, I got it. And she did. So people think differently about things. I think that what doesn't hurt is to be enthusiastic. I hope you've gotten that I'm a fairly enthusiastic person about the things. I think that's useful. I think liking what it is. I think liking people and working with people is a good idea. I think having those problems that don't work out being in the back of your mind bothering you all the time is a good thing to do. But I think that people have very different approaches. I can tell you another thing that it's not. It is not grades. That doesn't say whether you're going to be a good scientist or not. I think it's more dedication, enthusiasm, and a willingness to learn and, to some extent, to play.
My question, should I go ahead?
Yes, please.
Okay. Could you tell us the story of how you personally became interested in human rights? And could you give us some examples of how human rights have been integrated into teaching or discussions? Some interesting examples, yeah.
So I hope that you saw that the example I gave. I don't think I can really give another example because I'll be too long-winded. I have always been somewhat peripherally interested in human rights. I've been a member of Amnesty International for a long time and wrote letters. But it was only about 5 or 6 years ago that I was asked by the Committee on Human Rights of the National Academies if I would join it. And I like to joke that Nobel laureates are often told that they should sort of get off their butts and contribute something. And that would be my wife again. That is, in fact, it was an opportunity I didn't want to miss. And then when I was asked to be chair, and I've been involved with a group of really astonishing people. And unfortunately, we are working currently on 70 different cases worldwide and investigating an additional 50 cases to see whether they fit our criteria of serious human rights abuses and no use of violence. And this is only people that have a background in the social sciences, natural sciences, engineering, and the health professions. And unfortunately, it is virtually worldwide that there are these horrible things that happen. There are cases where academics, particularly engineers or scientists, are teaching school in homes because it's illegal for them to teach the students that they want, and they get arrested and put in jail for teaching. There are people that have signed petitions for human rights that are academics that have been fired for their jobs and then arrested. There are doctors that treat people who have been injured in riots, but because the government does not like those individuals, they arrest the doctors. So this is a problem everywhere, where people bring to others' attention. It doesn't have to be in terms of their scientific work that they are arrested or have other human rights. They could be just doing a blog or assembling or any of the major human rights problems. And I've become quite passionate about this and concerned, and it's a way I hope I can contribute.
Thank you. Please.
Hi. I was just wondering, not that I don't like the green color, but why do you have to insist that until you get the green light? Why when you're happy with the blue light?
So the blue light came from a quarren. Now the blue light from a quarren, that was a chemical reaction that was bioluminescent. So we had to keep, if you wanted to use that, you would have to keep adding the chemical for the reaction. You didn't just need the enzyme, you needed the chemical to do that. You could do that. There was a paper several years before ours in which people took firefly luciferase, the bioluminescent molecule that allow fireflies to produce light. And they were able to put that in a plant cell, and they prominently had the paper, a picture of the plant leaf, actually whole plant cell, on the cover of Science. The problem was because every chemical reaction made one photon of light, they had to put it in a drawer on top of photographic film for 24 hours. Using fluorescence, we can add as much substrate, blue light, as we want and see the green light coming out. So instead of a chemical converted to a light, we have light of one wavelength converted to another. Much bigger reaction, much faster. We can see things in real time.
I think this might be the last question put up here, please.
So I think Max Planck once said that science events is one funeral at a time. One funeral at a time. And I was wondering, how would you sort of encourage an undergrad who is looking at the flow cytometer or who's looking at, a fluorescence microscope for the whole day? Like, what's the inspiration for them to keep doing science?
When In 2004, I was elected to the National Academy of Sciences. And they have perhaps the world's most boring induction ceremony that anyone can imagine. Each of 70 people takes about a minute per person. Each person gets up on the stage, shakes hands with the president of the National Academy, has a picture taken with the president of the National Academy, walks across the stage, and signs their name in a book. Now, at first you think you're signing the book that Einstein signed, and then you later find out that the book was started in 1970, so that was not possible. In any case, that happens to 70 people. And you can imagine in the audience, everyone is there for one of those 70. So it's not a really exciting ceremony for most of the people. My daughter was 12 at the time, and I eventually made my way after this, thinking that she must have been bored out of her mind. And I went up to her and I said, look, sweetie, I'm really happy you and Mom are here to help me celebrate this. This is a nice honor, and I'm glad you're here to share it with me. But I'm sorry, I just want to apologize to you. This must be deadly for you. And my daughter looked at me, as she's often looked at me, that I'm the biggest idiot on the planet, and said to me, Dad, don't you understand about these people? And I said, what do you mean? What about these people? She had been listening to the two sentences that were used to describe everyone as they walked across the stage. And she said, Dad, every one of those people on the stage were the first person in the world to see or do what they did. I'm exceptionally proud of that answer. I think that's amazing. That is one of the things we do. We get to be the first people in the world to make these discoveries. They don't come every day. They're a lot of work, but they do come, and it's exceedingly rewarding. So that would be my answer to that.
Marty, that was a perfect end. And I think I'm just going to call for one more round of applause for all of you for being here, for Marty for taking the trouble to be with us now and for the last 3 days, and also to your exceptional team at McGill who've put this together so beautifully. Thank you very much.